In this video, we look at how DNA was found to be the molecule responsible for passing on genetic information from one generation to the next, and how it copies that information in order for it to be passed on to the next generation of cells. At the time that scientists were trying to determine what genes were composed of, they already knew and understood that chromosomes were made up of both DNA and protein, but they didn't know whether genes were exclusively comprised of DNA or protein. The general consensus at the time was that most probably genes comprised of proteins because of their relative complexity in comparison to DNA, which is only comprised of 4 different nucleotides. So scientists at the time didn't think that DNA could have that flexibility and variability to store large amounts of genetic information. To determine whether DNA was in fact the material that was part of genes, 2 scientists, Alfred Hershey and Martha Chase, in 1952, used the virus T2 that infects the bacteria Escherichia coli as a model system to address this question. The way that this virus infects bacterial cells is by attaching to the cell surface and injecting its genetic material or genes into the cell of the host, followed by the use of this information to generate more viral particles that break open the host cell, leak out, and infect other bacterial cells within the same colony of bacteria. This is a modified microscopic image of the virus actually attached to the bacterial cell, and you can see that it's injecting its genes into the host cell. The capsule of the virus stays attached to the outside post-infection. Hershey and Chase wanted to determine whether the genes that were injected into the host bacterial cell were made up of DNA or protein. So they developed a hypothesis stating that viral genes consist of DNA. In the experiment that they set up to address this hypothesis, they grew virus cells in the presence of one of 2 radioactive isotopes, P32 or S35. Since DNA contains phosphorus, only DNA would be labeled with P32, and since proteins contain sulfur and not phosphorus, only the proteins would be labeled with S35. So now you have the protein of the viral cells labeled with S35 and the DNA labeled with P32. So one sample in which the DNA is radioactively labeled and another sample in which the protein is radioactively labeled. These labeled viruses were then used to infect E. coli cells in 2 separate test tubes. The scientists hypothesized that if genes consisted of DNA, the radioactivity would show up inside of the bacterial cells. This can be determined by actually physically agitating and separating the viral particles from the host cells that are infected, that contain the viral genes inside of them, and spinning or centrifuging the cells. Since the viral particles are lighter than the bacterial cells, they would end up in the top layer called the supernatant. So based on the hypothesis that the scientists had, if the viral genes were composed mainly of DNA, then radioactivity would show up in the left-hand sample in the bacterial cells that are at the bottom of the test tube. On the other hand, if proteins were the main component in genes of the viral cells, then radioactivity would show up in the bottom pellet. After completing their experiment, Hershey and Chase found that radioactive protein was found in the supernatant of the tube, which consisted of radioactively labeled protein, and not in the pellet, which was settled at the bottom of the tube after centrifugation. The radioactively labeled DNA was found in the pellet, which indicates that viral genes that were injected into the host cell in both cases are actually made up of DNA and not protein. This was a key finding in the field that led to the discovery of DNA as the molecule that contains all information for life's complexity. Based on the conclusions from Hershey and Chase's experiments, Watson and Crick, who by this time had already proposed the double helical model for DNA structure, suggested that the existing DNA strands could serve as templates for the synthesis of new DNA strands. In this case, the nucleotides would be added to the growing new strands according to complementary base pairing. At the time, there were three hypotheses for how the old and new strands interacted during replication. One was the semi-conservative replication hypothesis, another one was the conservative replication hypothesis, and the third was the dispersive replication hypothesis. According to the semi-conservative replication hypothesis, the two parental DNA strands separate from each other. Each of the parental strands are then used as a template for the synthesis of a new strand. As a result, the daughter molecules consist of one old and one new strand. According to the conservative replication hypothesis, the parental molecule serves as a template for the synthesis of an entirely new molecule. Whereas in the dispersive replication hypothesis, the parent molecule is actually cut into sections and the daughter molecules contain old DNA interspersed among the newly synthesized DNA segments. Two scientists, Michelson and Stahl, designed an experiment to provide more information about these hypotheses. Their results supported the semi-conservative hypothesis for DNA replication. This explained how hereditary material is duplicated. Their experiments showed that each parental strand is copied in its entirety, but they still did not illustrate a mechanism for the process of DNA replication. It was when the enzyme DNA polymerase, which catalyzes the synthesis of DNA, was discovered that we gained some insight into understanding how DNA replication reactions work. Let's talk about some characteristics of DNA polymerases that are important for the synthesis of new DNA strands by the enzyme. DNA polymerase can work only in the 5' to 3' direction, meaning that if you consider this as the parental strand, DNA polymerase can synthesize or add nucleotides to a growing new DNA strand only in the 5' to 3' direction, which is anti-parallel to the parental strand. It adds deoxyribonucleotides to the 3' hydroxyl group of a growing polynucleotide chain to form phosphodiester bonds between adjacent nucleotides, which is indicated here. This leads to the release of a diphosphate, a water molecule, and energy, which is stored within phosphate bonds. Therefore, this reaction is considered to be exergonic, meaning that it releases or gives out energy. So how does DNA replication get started? If you look at bacterial chromosomes, they are circular DNA molecules. When DNA replication begins, a replication bubble forms in the chromosome, separating the two parental strands. This replication bubble grows as DNA replication proceeds in two directions. Similarly, in eukaryotic chromosomes, when DNA replication begins, replication bubbles are formed within the chromosome. If you compare bacterial and eukaryotic chromosomes, though, you can see that in the bacterial chromosome there's only one replication bubble, whereas eukaryotic chromosomes, which are much larger linear molecules, have multiple replication bubbles. The site where the replication bubbles form is known as the origin of replication, resulting in the formation of a replication fork, which is a Y-shaped region where DNA is split into two separate strands for copying, and this fork moves in two opposite directions. This is why replication is said to occur in a bidirectional manner. DNA replication begins when an enzyme called helicase binds to and opens or unwinds the double helix, separating the two parental strands. This forms the replication bubble that we looked at earlier. Helicase unwinds DNA by breaking the hydrogen bonds between base pairs. To prevent the separated strands from coming together again, proteins called single-strand DNA binding proteins, or SSBPs, bind and stabilize these single strands. Another protein called topoisomerase releases tension that is created by this unwinding action of helicase ahead of the replication fork. This prevents damage to the DNA double helix. Once the helicase separates the two strands of DNA, an enzyme called primase, which is similar to RNA polymerase, binds one of the single strands, called the leading strand, and synthesizes a short RNA primer for the benefit of the DNA polymerase. Now, why is this needed? DNA polymerase can catalyze DNA synthesis only in the 5' to 3' direction. In addition to that property, it needs a free 3' hydroxyl group to be able to add nucleotides to. Therefore, there is a need of a primase to synthesize a short fragment of nucleotides so that DNA polymerase can add to this already existing fragment. And thus the synthesis of a new strand begins when DNA polymerase binds the primer and starts extending it in the 5' to 3' direction on the leading strand, moving toward the replication fork. This synthesis occurs in a continuous manner without any break, which is why this strand is called the leading strand. So what about the lagging strand? Since DNA synthesis must proceed in the 5' to 3' direction, the second separated strand in DNA now has a problem because the replication fork is moving in a direction that's not compatible with the synthesis of DNA. In this situation, DNA replication occurs in a direction away from the replication fork and in a discontinuous manner, as we'll see on the next slide. Therefore, the second template strand in DNA is called the lagging strand. As the replication fork moves, it exposes more single-stranded DNA on the lagging strand. Similar to the leading strand, synthesis of a new strand begins when primase adds a short RNA primer that is complementary to the lagging strand. The difference is that now the DNA polymerase 3 binds the primer and synthesizes DNA still in the 5' to 3' direction, but away from the replication fork. It continues to do this for approximately 1,000 base pairs, after which a second primer is synthesized, and the process continues to synthesize a second fragment. So synthesis on the lagging strand occurs discontinuously in the form of fragments known as Okazaki fragments, named after the scientist that discovered them. DNA polymerase 1 comes in to remove the primer and fill in the gap with deoxyribonucleotides. The two Okazaki fragments are still disconnected because there isn't a phosphodiester bond linking the two fragments. This is taken care of by the enzyme DNA ligase, which comes in and catalyzes the formation of a phosphodiester bond between the two Okazaki fragments, making them continuous with each other. Simultaneously, at the replication fork, you can see that a third fragment has started to be synthesized, which will be joined to the second fragment. This process continues until the entire DNA molecule has been replicated. All of the enzymes that are involved in DNA replication are arranged in a very organized manner around the replication fork. This arrangement, this assembly of all of the proteins that are involved in replication, is called the replosome. You can think of it as one large multi-enzyme machine that is responsible for synthesizing new DNA. Let's take a look at the machine at work by watching a video about DNA replication.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
This concludes our section on DNA replication. At the end of this section, I'd like you to be able to answer the questions listed on this slide. Thank you.